Hey guys, today we'll learn the structure of DNA and RNA in a very, very simple manner. So, what is DNA? So, first of all, the very fundamental of life depends on DNA. Why is a monkey different from us or an onion different from a monkey? Now, in a fertilized egg, like in an ovum, okay, let's take zygote. So, in a zygote, the genetic information, the genetic material that is present in the nucleus has the complete program for the organism. So what particular organism that needs to be made? So it has that complete program. And that program is stored in the DNA. So we can say that DNA is, it kind of has the master plan of life. And DNA expresses itself in the form of RNA. So the master plan is in the DNA. Master plan is in the DNA, and the functional copies are in the RNA. And what does RNA do? RNA actually forms proteins. Now, what are proteins? Now, proteins are made up of amino acids. Like, these are amino acids, and this is a protein molecule. So proteins are of many types, like structural proteins and functional proteins. So if you talk about structural proteins, they make structures like skin, bone, hair. So what kind of proteins make skin? These structures, a few examples are keratin, collagen. So these are structural proteins. And there are certain proteins which are functional proteins, which do certain functions like hemoglobin, which carries oxygen, enzymes, which are biocatalysts, insulin, which is a hormone. So these are functional proteins. So in a cell, the DNA expresses the RNA, and the RNA will make certain proteins, and these proteins will determine the structure of the cell and also the functions. So structure and the functions of the cell. So it means that the DNA determines the RNA, DNA determines the RNA determines the type of protein that needs to be made. And these proteins determine what type of cell has to be made. The type of cells will determine the organs that need to be made, and the organs will determine what type of systems that needs to be made, and those systems will determine the functions forming the organ system, and the systems will determine what kind of organism it has to be. So this is a simple mnemonic again, which will help you understand the levels of organization. So this is how the whole system works. So now let's get back. What is DNA? What's a DNA? So what's a DNA? So think about it and tell me what a DNA is. Okay, some of you would have, just given the full form of DNA, which is deoxyribonucleic acid. Okay. So now, before we get into what a ribonucleic acid, deoxyribonucleic acid is, DNA is a polynucleotide. Okay, DNA is a polynucleotide. Like supposing if we have this strand of DNA, it is made up of a lot of nucleotides. Imagine these are nucleotides. Like, these are nucleotides. Like how we saw amino acids make a protein molecule, like that, the DNA molecule is made up of nucleotides. Now you must be wondering, what's a nucleotide? What's a nucleotide? Okay. Now what's a nucleotide? It is, if we have lots and lots of nucleotides, we can actually make lots and lots of nucleic acids. Nucleic. So mainly there are two nucleic acids, DNA and RNA, and both of them are made of polynucleotides. So a nucleotide is the basic building block of nucleic acids. So remember, if we have a lot of nucleosides, we can make a lot of nucleic acids. We can make a lot of DNA and RNA. So to make DNA synthesis, like to make DNA, we need some raw materials. So those raw materials are the nucleotides. Now, what is the nucleotide made of? So the raw materials that are required to make this nucleic acid are nitrogenous bases. So what do you mean by nitrogenous bases? It contains, from the word, see, nitrogen. So these bases, they actually contain nitrogen. They contain nitrogen. Okay. So these nitrogenous bases, they are actually carbon rings which have nitrogen bases. So what are the different types of nitrogenous bases? They are adenine, guanine, cytosine, and thymine. So there is another base which is called uracil. This is called uracil, but this uracil will not be found in DNA. It will be found exclusively in RNA only. So we'll learn more about it later. So remember, the nitrogenous bases are adenine, guanine, cytosine, and thymine. So these four bases are found in a DNA molecule. But this is not enough. To make DNA, the nitrogenous bases are just not enough. DNA or RNA, the nitrogenous bases should be fused with sugar. With sugar. Okay. Now, if you see sugar molecules, let me use this part of the board. So if you see the sugar molecules, there are lots of sugar molecules, but in this case, we will be talking about pentoses. Now, why pentoses? Because they have 5 carbons. Okay, they are made up of 5, they are rings. They are carbon rings, and they are made up of 5 carbon sugar. Therefore, they are called pentoses. Now, specifically, the pentose that we are talking about is the ribose sugar. So remember, ribose sugar. So you saw deoxyribose sugar, right? You saw here that DNA is deoxyribose. So here you go. So that ribose sugar is nothing but this carbon ring. So remember now, let's fuse the sugar, okay, with the nitrogenous base. So this is the nitrogenous base. So adenine is A. Okay, let's try to make a strand here. So they are attached to the side. So let's say adenine, guanine, okay, cytosine, C, and then thymine. But then, how are we going to attach it? We need a backbone, right? Now, you need a backbone to attach these, the ribose sugar and the nitrogenous bases should be attached to each other. So for that, there is a backbone called the phosphate that is tied to this. So this is a ribose sugar. These are bases, and this is a phosphate group. So this is the phosphate group. So this phosphate group will actually make the backbone of this strand. So can you see this is a single strand? It is made up of a phosphate group, a ribose sugar, and a nitrogenous base. And that nitrogenous base can either be adenine, guanine, cytosine, or thymine. So this is a nucleotide. This is a nucleotide. And this nucleotide, when it combines, when it gets added up to more nucleotides, they are called polynucleotide. Now, DNA, you must have seen DNA is a double helical structure. Now, there are a few enzymes which help in making that double helical structure. If you see, this is one strand. There is another strand that needs to be made here. And there is one rule. There is a rule for this. So there is a base pair rule. So if it is adenine, it will pair up with a thymine. It will pair up with the thymine. So here's the base, and then you have the ribose sugar, and you have the phosphate group. Okay, so guanine will always pair up with a cytosine. So you have the ribose sugar, okay, and this phosphate group will actually kind of it makes the backbone here, and it's the reverse. It's the reverse. So cytosine, so guanine. So it's like ATCG. So look at this, ATCG, ATCG. So it's, you have to remember this, memorize this. A and T will, pair up, and C and G will pair up. And the thing is, the bonds that you see in between are hydrogen bonds. CG. So here you go, here you have a double hydrogen bond, here you have a triple hydrogen bond, double, and it goes on. So here, this is how it makes a double-stranded, molecule. It's like this. It makes a double-stranded molecule, which are complementary to each other. So the bases are complementary to each other. So A, T, C, G. So here it is C, so it should be G here. And here it is, and there is a triple bond, triple hydrogen bonds. These are hydrogen bonds. You will be learning about it later on, not now. So the phosphate group here, the ribose sugar. So this is how the whole structure is made. So T, so here it should be A. So this is a rule. So this is called the base pair rule, ATCG. ATCG. So you see this picture clearly shows how the pairing is done. So if you see a picture like this, then you must understand. If you see a picture like this, then you must understand that this is the sugar phosphate backbone. This is the sugar phosphate backbone. This is the sugar phosphate backbone. And what you see in the middle, the rungs like a ladder, this looks like a ladder, right? So these rungs are the nitrogenous bases which are paired to their complementary bases. These are nitrogenous complementary bases. So they complement each other. So adenine will always be paired with thymine. Guanine will always be paired with cytosine. Cytosine, guanine, thymine, adenine. Either ways. So this is a DNA molecule. So this is one strand and this is another strand. So together they are a double helical structure and they are a little twisted. So the complementary bases are ATCG, and these are not simple but twisted strands. I hope that is clear. Okay, while DNA is a double-stranded structure, it's a double-stranded structure. RNA is always a single-stranded molecule. It's always a single-stranded molecule. So DNA is found in the nucleus. Now, if you, let me show you a picture. I have a nice picture here. Yeah. So if you see, let's say you have your grandma's secret recipe book, which is kept really safely. You can't take it or shift its place, but can copy the recipe from it. Exactly like that. You have the DNA which contains the code. You know, the DNA which contains the code to build the proteins, and the RNA uses these instructions to build all the proteins a living organism needs. And there are three types of RNA. They are messenger RNA, otherwise called mRNA, ribosomal RNA, otherwise called the rRNA, and transfer RNA, which is also called the tRNA. So RNA is called ribose nucleic acid because it has an oxygen in it. Can you see here? The oxygen here. Whereas here there is no oxygen, therefore DNA is called deoxyribose nucleic acid. So now let's do a little bit of recap of the differences between a DNA molecule and an RNA molecule. So DNA is always a double-stranded. Can you see the double-stranded molecule structure here? So they are double-stranded. So this is how it is. Okay, let me clear my diagram. So this is a double-structured or double-stranded molecule, and RNAs are always single strands. Okay, they are not double, they are always single stranded molecule. And DNA is found in the nucleus only, but RNA are found in the nucleus and also in the cytoplasm. So DNA has nitrogenous bases such as cytosine, guanine, adenine, and thymine. And the nitrogenous bases of RNA are cytosine, guanine, adenine, and uracil. So there is no thymine in RNA. Okay, so these are the basic differences between RNA and DNA.